Today I will be talking about building TMS tools for selective neuroengagement from the ground up. And I have parsed this title into 3 lines for a reason. First, building. This means that it's stuff that we're doing right now. So a lot of the work that I'm going to show is work in progress. And I really intended this to be to share the excitement of some of the new technologies that we're developing and what they can offer us. Selective neural engagement, selective target engagement, that's a very important part of the motivation of a lot of studies and funding these days. How can we improve our tools to have more selective effects on the brain? And finally, from the ground up, that emphasizes that we're looking and we're doing work at a very, I would say, low level in the sense that we're really designing the devices which we use for TMS, the pulse generators, the coils, modeling at the level of individual neurons in the brain. So we think of this as really building up our understanding and technology for TMS from the ground up. Want to give my disclosures. We work on technology, so we have pretty extensive collaborations with industry, including various companies that work on magnetic stimulation technology. We have received some research funding from them, and we have worked with them to commercialize some technology. And also we have received extensive support from the NIH, for which we're very grateful, and that has enabled the majority of the work that you're going to see. And we have received funding from other foundations and organizations as well. And importantly, I want to acknowledge the people who have done most of the work that you're going to see. And actually one of them, my former PhD student, Zita Deng, is now here at the NIH, and it's great to see him. There are many other lab members. I have highlighted 3 of them who have been really the central effort behind what I'm going to show you. Luis Gomez, Stefan Goetz, and Amanda Vera. And also want to acknowledge my faculty collaborators, which include Dr. Holly Lisenby and Bruce Luber, who are both here at the NIH now. So I won't spend much time introducing TMS. I think you are familiar with that, and I think most of the audience online is familiar as well. TMS is a technique that uses strong magnetic pulses generated by this TMS device that are sent to the brain, and there they induce an electric field which results in changes in neural activity. So TMS is really a way of inducing electrical stimulation in the brain using electromagnetic induction. So it's good to keep in mind that TMS is fundamentally electrical stimulation. It's just delivered in a way that's tolerable to awake subjects and patients. You're probably familiar also with the applications of TMS. And here I have a brief summary. There are 3 main categories: research applications for understanding brain function, therapeutic applications in psychiatry and neurology that are, the list is expanding. We know that TMS is FDA approved for depression and also for pre-surgical cortical mapping. It's also approved for migraine, although one may argue that that's more like cranial nerve stimulation, possibly. But the list of many other disorders that are being explored experimentally at this stage and may in the future establish therapies as well. And finally, application enhancing performance subjects. So, these applications, the applications can be as good as really our technology and understanding of mechanisms is. So what are the limitations and what can be done about that? Well, here I pulled an image from the web from the Mayo Clinic, which is obviously a great clinical institution, but when you consider how they advertise TMS, you can see some problems with this picture. The TMS coil, indeed, is placed in the scalp and looks like a real TMS coil, but then it looks like it emits this pretty narrow beam of magnetic field into the brain, the target that's quite deep, and it's kind of a focal activation there. So unfortunately, So the idealized view of TMS is far from what actually happens. And to optimize for the TMS, we have to have an appreciation of real TMS rather than this idealized picture. So I here started adding and correcting this figure to emphasize what actually happens. So first of all, the strongest stimulation and activation happens in superficial brain regions because the field attenuates in depth. We can characterize that area of activation with some depth and spread, which gives you the overall focality of stimulation. But of course, then you have field that is, it's not the beam, you have field around it that is weaker, but it still, covers some extent, and there are probably subthreshold effects in these regions. Then within the target and also in the surrounding tissues, there are various neural populations that you may be affecting, right? And you may want to target just one of those. Then there are transsynaptic activations. The brain is interconnected, so when you stimulate these neurons, you stimulate fibers and tracts that go to other brain areas and so on. And then beyond the brain, actually in the scalp, you have nerves and muscles in the scalp. You stimulate those. The field in the scalp is stronger than in the brain, right? Then the coil, because you send very strong energy in the coil, it also vibrates. So you can actually feel the vibration. So even that's a sensory stimulation that goes to the brain. And because of this vibration, the coil, if you hear TMS, it makes a pretty loud sound. So you have vibration that's transmitted both through air and through the bone of the skull, and you hear that sound, both bone-conducted and air-conducted sound. So all of these kind of additional activations, I lump them in this both saying we have all these unintended effects that occur during the administration of a pulse that are beyond our ideal desire to maybe stimulate one of these neural populations here, the focal spot of the coil. Right, so that kind of looks somewhat disappointing, and it is a result of various fundamental physical constraints of TMS, but not all of it is fundamental. And we have been trying to really rethink the design of TMS, of how we can really drive real TMS closer to ideal TMS. And in part, it's understanding how these processes work and also eliminating some of these extraneous effects that we don't want. So I have a list here of how I think of functional selectivity of TMS, which is what we want to accomplish, and factors that contribute. And first of all, probably the spatial focality of the electric field that's determined mostly by the coil. Then the temporal characteristics of the TMS pulse can also affect how you recruit neural populations. Of course, the repetition parameters of pulse also can interact with the dynamics of larger populations, and they have significance in what you affect and how. Then there's neural connectivity. You can stimulate the superficial brain area and then you can stimulate deeper areas by that. Right? It's a very promising way to target deeper brain structures. It matters what the brain is doing while it stimulates, what the context is, and I think we should take more of that into account. And finally, there are these ancillary effects, auditory stimulation, somatosensory stimulation, that are not fundamentally a part of the process. So if we can kind of minimize them, right, we can have a more selective stimulation process. So in this presentation, I'm going to give you really this kind of brief overview of some of these technologies that we're developing to address to improve TMS along these lines. That would be along the lines of spatial targeting related to coil design and pulse shape targeting related to the TMS waveform, and also the effect of actually the sound of TMS. So first starting with coil design. In this figure, this is the result of his work for his PhD thesis, where we put together kind of a list of most of the coils that could be published or conceived or proposed in TMS, and wanted to evaluate how focal and deep can TMS be at the same time, right? We really want something that's focal and also can go deep, but what are the limitations? So here we simulated this, in this case, 61 coils, and it resulted in this plot that has been cited quite frequently because it actually shows a very kind of very definitive trade-off between the spread of the field, that is the kind of tangential focality, how much of the brain you stimulate, how much of the brain surface you stimulate, and then versus the depth of stimulation, how deep the field penetrates. So you see there, all of these coils are pretty much lined along two curves. The lower one, which is the better one, right, for given depth, it gives you more focality. This one corresponds to the conventional figure of 8 type coils, basically 2 loops of wire. And then you have, when you have larger coils, you go up this curve. When you have smaller coils, you go down this curve, but you're always on this curve. And all kinds of tricks that people have proposed didn't move them. They didn't budge from this curve. I mean, they may get worse than this curve, but not below this curve. So I personally thought that that's it, that we should reach the limit here. But then we got this new postdoc in the lab, Luis Gomez, whose PhD is in computational electromagnetics. And he's very bright, and the kind of tools that he has from computational electromagnetics allow us to take a very different approach to coil design, which he had started working on in his PhD at the University of Michigan. He's now continuing in my lab too. And it is not to play with, come up with configurations and try them out, which is what people have done before, but actually computationally synthesize this coil so that it optimally meets our requirements, our specifications, which makes a lot more sense, right? You're asking basically, fundamentally, what is the best thing to do for targeting, with these parameters, rather than you think how it may be, just, coming up with drawing your design. Basically, computers are better at electromagnetics than we are, because they can, take all of the relationships into account, and our brain can't really do that. So the way this approach works is you specify some requirements, where the target is located, what should be the field direction there, what is the stimulation threshold, you can estimate that, and that's not, that's something that's scalable, so it's not an issue. How strong can be the surface field relative to depth field, that's a constraint you can put in, and how much energy we can practically use for the coil. So you can put all of these constraints, then you choose whether you want a flat coil or a curved coil or a cap coil. So you choose kind of the overall shape of the coil, like it's a plane, and you also choose what model you want to do the optimization. You can use a spherical model, which is what we use for these evaluations, or you can use a realistic head model derived from MRI. So the first mention of MRI, so that's an interesting use of multimodal techniques, where here we actually use structural MRI to design coils for TMS. So once we have made these choices, then the computer algorithm basically breaks down the surfaces into these spatial harmonic modes, and then weights them in different combinations and goes to extensive search for combinations to meet in an optimal way these specifications. For example, optimum may be getting all these specs with a minimum stimulated volume of the brain, so with a maximum focality. So it iterates to this loop, and you run that on a supercomputer. It may take a few hours or a day in the worst case, but that's no problem. Now we have supercomputers. Everybody has one at this point on their campus. And then it gives you code design. And I have here, it's like a cliffhanger, because we are in the process of it. This is fresh, fresh from like as of last week, what's going on. So we are, this may be important technology, so we're about to submit an innovation disclosure. So I'm not disclosing the actual quotes we're getting, but hopefully we'll be publishing that soon. But I can show you on this curve what we are getting in terms of performance. And we think that this is the fundamental limit, actually, when we without constraints of the coil energy. So it looks like we can be a factor of 2, not with this logarithmic plot, but you get approximately a factor of 2 from here to here for the same time. So the point is that for given depth, we may be able to increase focality as measured by the spread of the field by a factor of 2, which I think is pretty good. I mean, I think it can be meaningful. A factor of 2 in surface area is, can be significant. And actually for deeper coils, that are generally non-focal, but there you have So I'll put this away in case it's in the way. And as I said, this is fundamental limit. So practical coils, I think we can get close to that. That's what also the practical energy constraints. So that's a little kind of peek into something exciting that we're working on, and that may result in a different, in a different category of coils that have better characteristics. And also, recently, the lab of Dr. Vistil Maniemi, who is speaking here at the NIH soon in this seminar series, they have done some great work also in that direction of computational optimization of coils. So really the field is going in that direction right now, and apparently we can do some things that, better than what is the state of the art now. So the next topic, obviously this is, the first two topics I'm going to cover relatively quickly, and the third topic will be a bit more extensive. My next topic is on our efforts to build devices that are quieter. So why is that an issue? If you have experienced TMS as an operator or as a subject, you know that it is loud. I mean, you have to wear earplugs. There are, safety guidelines on that. You have to protect hearing. The operator also is supposed to protect hearing. You can hear often the device down the corridor, even with a closed door or in neighboring rooms. If you quantify the noise, it can be over 100 decibels. At maximum output, it can be like 120 decibels. So that's loud. That's there are various comparisons, like like leaf blower or jet engine and some distance. There are many kind of comparisons. It's basically very loud sound. It doesn't sound as loud because it's very brief pulse, but nevertheless very strong sound. So it's not just a nuisance. It's actually a major compound of TMS because what you do is you activate the auditory system synchronously with each pulse that you deliver to the cortex magnetically. And if you look at what happens in the brain, and this is, again, multimodal, TMS fMRI in this case, you can see on this picture, you stimulate a lot of brain through the auditory perception. On this figure here, you see, again, this is TMS fMRI, intensity of pulses delivered to frontal target ranges from 100% of motor threshold to, from 80% to 100% to 120% of motor threshold. And you can see that under the coil, below motor threshold, you don't have much activation. At 100%, you see some activation, and then at 120%, you see more activation, which is what you would expect from ideal TMS. But look at what's going on. Auditory cortex, brainstem, they're all lighting up, even at lower intensities, and that's because you have a loud clicking sound, and actually it's worse in the MRI scanner, because you have the static magnetic field, so this amplifies these forces. Also, the bore reverberates, so it's like this, reverberation chamber, so it's really loud. And when you look at this, you can't really say we're focal. I mean, you can replicate these effects with sham, right? And you can say, well, I'll have the same effect, but it doesn't mean that you can just subtract them out. The phrase is obviously non-linear system, that you can't just be, replicating something, just subtracting that and saying, okay, that didn't have any other effect, I just subtracted it out. So, so sound is a real issue, and it can, even auditory stimulation by itself can modulate the brain. If you stimulate somebody with auditory stimulation temperatures, there'll be actually neuromodulatory effects that are lasting. It complicates blinding in studies. Obviously, it's a risk to hearing. If the hearing protection fails, some patients may be sensitive to it. So it's really something that we'd rather not have. And when you think about it, it's an acoustic field. It has nothing fundamentally, physically to do with the magnetic field. They don't have to come hand in hand, right? So you should be able, theoretically, to design a TMS device that makes no sound, simply by not allowing it to produce any mechanical energy, only having magnetic energy come out of it. And that's what we are trying to do. So we call the approach quiet TMS or QTMS. We have a lot of TMS with modifiers in front. So it's kind of, could be also QTMS. So it's a two-pronged approach. The first part of the approach, which is probably the less intuitive part, is that you'll think, oh, you just make it quieter coils. It's not just the coil, because the problem with conventional TMS is the energy of the pulse that you send to the coil. You send it sinusoidal pulse, and it is centered in it's in the range of 2 to 5 kilohertz, which is smack into the best hearing range for humans, right? We're rated hearing these frequencies up to, for young people, 20 kilohertz. For older people, it kind of rolls down a bit, but that's smack into where we hear the best, right? So we have to start, so one point is let's get out of this range. So what we propose to do is move the pulse frequency from 3 kilohertz by a factor of 10 to something like 30 kilohertz. So then you're outside of the human hearing threshold. So it's a good start, right? At least conceptually, if you keep the energy in that frequency band, you shouldn't hear much of it. Now, why is that not trivial? It's not trivial because this means making the pulse, the TMS pulse, 10 times briefer to have 10 times higher frequency. So making the pulse 10 times briefer, because we have strength duration curves in neurostimulation, this means that basically you have to increase the amplitude of the pulse by a factor of 10, approximately. And that is technologically challenging. And actually with conventional devices, making such a brief pulse is also challenging, but we have already technology that can do the briefness, but the amplitude is a challenge. So we're going to address that by building a device that produces these briefer pulses and uses higher voltage and higher currents to get that factor of 10. You can split it between current and voltage, basically, with appropriate coil design. Interestingly, the energy that's needed is about the same. We have this 100 joules as a kind of approximate threshold. That doesn't change. In fact, it's theoretically slightly lower for briefer pulses, even though the pulse is high amplitude because of the brevity, it's lower energy. And the second part of this two-pronged approach is to build a better coil. And the purpose of the coil, reasonably, is to make sure that the magnetic energy that you deliver to the coil is not converted to mechanical energy, or rather a minimum amount is converted. And not only that, but the conversion is linear. That is, you don't have down mixing of the frequencies from high frequencies to low frequencies because of mechanical vibration modes of the coil. So you really, whatever energy you end up emitting, is still at this, 30 kilohertz rather than 3 kilohertz. So that's a bunch of mechanical engineering, and it turns out that the best way to do it, you have multiple layers within the coil. You have a very stiff winding, and then you have these different layers, viscoelastic layer, elastic layer, and then another like stiff casing. This actually comes from literature on machine design for quiet machines and with appropriate adaptation. So when we combine these two approaches, we hope to have this, kind of combined significant improvement in the sound that's outputted by TMS. And that's an actively ongoing project that's funded by a BRAIN initiative, R01. We're in year 1. However, before this R01 started, we already had demonstrated a scaled-down prototype that still used conventional voltages from the order of 1 kilovolt and kind of scaled the whole comparison appropriately so you can see the benefits of it, even though you can get to threshold. It's basically sub-threshold still. And with the new device, we have managed to go to threshold levels. So at the sub-threshold levels, we scaled everything. We scaled conventional pulse to be at the proper sub-threshold level, and our quiet TMS pulse were at sub-threshold levels. So I have this matrix, comparison matrix, where this is the acoustic waveform that you record with a microphone coming out of the coil. So here on the upper left plot, you see the acoustic waveform coming out for conventional maximum coil with a conventional maximum duration pulse of 300 microseconds. Then to the right, you see the conventional maximum coil, but with a brief pulse, 45 microseconds with the existing devices that we have specialized devices, but even we can get 30 microseconds with them yet. But it's still much briefer, right? And then you see that the amplitude of this waveform is reduced significantly. Then at the bottom or in the middle row on the left, you see the quiet TMS coil that we developed with a conventional pulse. And again, it's very weak signal. And then finally, we combine the brief pulse with the coil. We have an even briefer and shorter pulse. And the plot here on the bottom right summarizes that. The loudness is given in decibels, and that's relative to the, essentially, the auditory threshold. There's a way to scale that and compute that from the waveform. That's not from humans, that's from computing that from the acoustic waveform. So you see the conventional device, and then we see our prototype with all the features together. It's in the blue bar. So you have 19 decibels, which is almost the factor of 10 reduction in the loudness. So that's already quite remarkable, and we're going for the plan to to go for 40 decibels at least reduction, which is a factor of 100. Now I have here waveforms that I will play, but we figured out that you can only play them actually if you're in slide preview mode. And I don't know how well they play over WebEx because they'll have to go through loudspeaker in the microphone. So I apologize to the viewers on WebEx, but I'm going to give a little demo. So this is conventional TMS sound. You may recognize that it is TMS, right? Clicking sound, that's what TMS trains sound like. Okay, so now let's hear quiet TMS. And that's for matched. It should be matched in terms of percentage relative to threshold as the previous pulse. Does anybody hear? Do you hear this faint clicking? Yeah, the young people, I guess you're all young. So when I have given presentations to in a boardroom meeting of older CEOs, they're like, where's the sound? I'm like, you can't hear the faint clicking? Like, no, I'm like, that's perfect, This should be our subject for the study. We're going to, so after we build the device, which we're now developing, it's a pretty challenging engineering project, but we think it's doable. We'll test it in humans. We selected relatively young subjects. Maybe that was a mistake. So, but it should be 10 times quieter than this. So,
Is there any difference with the surface stimulation or the sensory?
That's an interesting question. So, so the question is, does that interfere with the sensory stimulation in the scalp? And we have done some studies with briefer versus longer pulses, but we haven't done such brief pulses. And it is possible that sensation may go down. There's some theories to that effect. And even if that doesn't happen, I don't think it will go up, basically, because the time constants of cranial nerves and nerve endings and sensing nerves, it seems that they tend to be generally a bit longer or the same as cortical neurons. So I don't expect it to get worse. It will be very interesting. I'm very curious to experience what that would feel like. And the only way we'll figure that out, that question came up from the reviewers of the grant, and we said, well, that's what we can hypothesize, but ultimately we can't test that because we don't have a device. So we'll know that by the end of this project. And now I'm moving on to the last component of the presentation, which is the most extensive. The previous two were basically previews of this technology that's in development to kind of whet your appetite. And I'm going to share here results that are a bit more kind of closer to publication. And that all has to do with, or already published, that all has to do with how we can leverage and understand the effects of pulse waveforms on in TMS, on neurostimulation. So first of all, for those of you with background in TMS, you may be familiar that conventional TMS devices typically offer you one of two waveforms, or some of them offer you both. The typical waveforms are monophasic waveforms, where the pulse is this kind of monophasic cosine pulse, and biphasic, which is, again, a cosine pulse that has a full period of oscillation. And the biphasic pulses are the basis of essentially all repetitive TMS devices, including those used for therapeutic applications. Some devices, actually just the MagVenture devices, I think, and maybe now some other ones, offer also this half-time pulse, which is sort of in between. There's a pulse in the negative phase. So even with this limited amount of pulses available, there have been quite a few studies that have shown that the pulse waveform matters. And one very interesting result of these studies is shown here. It's from Martin Sommer and colleagues, published more than 10 years ago, where they compared these monophasic pulses with half-time pulses and biphasic pulses. And they also changed this within multi-cortex. And they also changed the direction between flowing posterior anterior, PA, so the induced current flows from the back to the front, or anterior posterior, which is the current flowing in the opposite direction. And you can see here that the latency of the motor evoked potential, which is the finger twitch that you get when you stimulate multi-cortex, is approximately the same for all of them, around 24 milliseconds, except for the monophasic pulse when pointed from anterior to posterior. So for that direction, the latency is significantly higher, by almost 2 milliseconds. And that's a big deal. 2 milliseconds, that's about a couple of synaptic lengths, because going to one synapse takes about 1 millisecond. So it's very reasonable to interpret this as this pulse with the monophasic pulse with anterior-posterior orientation of the field is stimulating a different neural population than the other pulses, and this signal then has to travel through a couple more synapses to then get to the output neurons from multi-cortex and get to your finger. So it's a sign of, even though the field distribution is the same, just the pulse shape combined with the direction of the field provides preventing some selectivity of what neural populations we're stimulating. And in that sense, it provides this additional selectivity. So that's very interesting and potentially important. Additional degree of freedom, or degrees of freedom really, that we have in controlling what gets activated in TMS. And yes?
Sorry, just
another question?
Probe on that. Does it really provide us more selectivity? I mean, the latency was like a 10% difference, but it was still accrued, right?
So I wouldn't think about, so the question is, does that really provide much selectivity? It's just, say, a 20, or 10% difference in the latency. But this is, we should not think about this as percentage, which I think the proper way to think is synaptic distances here, because most of the reason that it's 24 milliseconds because it takes time to travel from your brain to your finger. So that's, we're talking about stimulation of cortex. And 2 milliseconds within the same cortical area, that's a lot. You're really stimulating, you have to travel through 2 synapses. So you're stimulating something different, a different population in the cortex. So it is significant. And actually that significance is really highlighted by studies again by Martin Sommer and by some other groups that I have 4 kind of thrown here as examples of exciting results that show that actually makes a big difference when you do neuromodulation with repetitive training. So for example, in this study, the one on top, they stimulated at 5 hertz with monophasic pulses in either posterior or anterior posterior, or biphasic pulses with one or the other direction. And when you see what happens to the MEP, that's really fascinating. 5 hertz is known to be an excitatory frequency, so the biphasic pulses that are conventionally used for RTMS produce, reasonable excitatory effect. The MEP increases by a factor of 2 approximately for either direction, and that makes sense because they're kind of bidirectional pulses. And that's what we normally get in studies with RTMS. But now look at what happens with the monophasic pulses. The posterior anterior pulse gives you this massive excitatory effect that's like almost 5 times the MEP increase. The MEP increases by a factor of 5, whereas changing the direction with the monophasic converts that to mild inhibition. So the biphasic then appears to be almost the summation of the two. You have this intermediate effect. So clearly, in this case, by using monophasic pulses with appropriate orientation, you can produce stronger and more selective in terms of what you're trying to get in terms of neuromodulation effect. And there, I won't go into detail into these other studies, but basically, pretty much any study that has looked at monophasic versus biphasic pulses seem to show that monophasic pulse, especially usually like a given direction of the monophasic pulse, produces stronger and or longer lasting neuromodulatory effects. So that again indirectly indicates that we're modulating the cortical circuits in a different way when we change the pulse waveform. So the first question is, what's going on? Like, where do these differences come from? And the field of TMS, really, we haven't had many good tools. It's really interrogating cortex at the level of individual neurons and how they get activated by TMS. And I think now with the proliferation of exciting tools and imaging tools in neuroscience, I think there are lots of great opportunities to explore that. And one of the opportunities that's now afforded by computational methods that are available is to actually do simulations. And that doesn't mean that they should go hand in hand with experimentation, and I'm going to talk about this later. But let's see what we can do computationally. Well, first of all, again, starting from a structural MRI, yet another use of multimodal techniques in TMS. If you take a structural MRI of the subject, you can then construct the 3D model of their head, including the different tissues that are relevant, like the brain, gray matter, white matter, CSF, scalp. And now there are packages available online that you can actually use. So it's very user-friendly at this point, techniques that should be more widely adopted. And I think that's starting to happen now, because it's really easy to do now. So with this technique, we can create a 3D model of the head, and we can simulate based on this electric field, right? And that has been done in the literature for a while now, and good, that gives you the electric field, but that doesn't tell you, for example, there will be no difference between the monophasic pulses going in fact, there will be no difference between biphasic or monophasic going in the direction of monophasic. All these different conditions that I showed you will give you exactly the same electric field map, because the coil is the same placement. So that doesn't tell you much. What really is different is how these field characteristics interact with the complexity of neural populations in the brain. So we have to start modeling that to really get an insight into what is the mechanistic basis of the different effects of different pulse shapes. And that's what we set about to do. So in addition to making these realistic models, which is again now relatively mainstream, we set about to differentiate different layers in the cortex. So you can see a cross-section here of our cortex. You have the layers from layer 1, layer 2, 3, 4, 5, and 6. And then we populate these layers with neural models that are pretty realistic. You can see one rotating down here. And you can see what the brain looks like, at least for one layer, when it's populated. Obviously, that's not the real density of neurons in the brain. It's about 140 times less than the actual density of neurons. But, you can increase that density if you wish, if you have the computational resources. And already with this density, it gives you a lot of detail into what happens in the cortex. So you can think of each neuron representing kind of a patch of neurons around it relatively well. And again, that you can, depending on computational resources, you can increase that. The neurons are based on a relatively recent data set, extensive data set that's published from the Blue Brain project in Europe that's headed by Henry Markham. So Markham's group did a really elaborate work in rodents at characterizing electrically cells and then also documenting their morphology and creating neuron models. Right, neuron is a simulation package for neural simulations. So we took these neural models and we adapted them to humans because these are from relatively juvenile rodents. And a lot of things are similar, like we're all mammals, but there are some differences. For example, we had to add myelination, and we had to scale the cells, and there were other adjustments that are all done based on well-understood basic either biophysics or neurobiology. It's not like we're tweaking them to give us what we want from TMS. We're really just building models based on what we know about how neurons are. And then with these populations, you can, you have E-field simulation, and then this E-field that stimulates each of these neurons with the local characteristics of the electric field. And then you can map activation of neural populations for each layer. And even within layers, you have different neural populations. So here you can see what we get out of the simulations. We can get this cortical map where now for each layer, you don't see the layers, just one layer here, but we'll see the layers later. You can get a map of the threshold for activation, right? So normally in TMS you do like a model threshold titration, you get one threshold, that's your model threshold. But here we're getting for each neuron, we're getting a threshold. So now it's a threshold map that we can do all kinds of things with. This is a library of the neurons that we model within each layer. So obviously within each layer you may have more than one type of neurons. We selected for this particular simulation, for simplicity, we selected the neurons with the lowest thresholds within layer. And you can add, and we have other models that can be added. And this ended being the most important ones, as you'll see, are layers 2, 3, 4, and 5, where in 2, 3, and 5 you have pyramidal cells, and in 4 you have interneurons, which the lowest threshold are large basket cells. So let's see the first thing that we got out of this kind of quantitative results. Well, you can ask, how do the thresholds for activating different layers compare against, the other layers? And these results you see for the different layers going from 1 all the way to 6. And you see this is a bunch of different, all these different monophasic, half-line, and biphasic in a different directions that we saw previously in the experimental study. We threw all of these in for fun. You can focus on, one particular color. But you see that the trend is pretty clear. And the trend is that layer 5 has the lowest thresholds, followed not too far behind by layers 4 and 3, 2. So layer 1 is 5, there are these cells that are way up there. They probably are very unlikely to be activated. Layer 6, especially given that it's deep, right, and the field is weaker, it's also probably unlikely to be activated. So this seems to be the layers that we should be focusing on, probably. And I wanted to point out that for those of you who actually appreciate the pulse amplitude in terms of amperes per microsecond, which is one way in which you quantify pulse amplitude, for example, the Mac-Pro device will give you that value as a measure of amplitude. These values are actually, like the lowest value here, they're on the order of 60% of the conventional device output. So, unlike some other studies with more simplified models in the literature, we have managed to get something that seems quite realistic in terms of thresholds, in terms of abscess thresholds, which is good, right? And that we had to do this myelination and a bunch of these kind of adjustments that are appropriate, and that really affected the threshold. So we think that we have pretty good models, and now we can play with them. So we like to, one way in which we like to play with them is to see how they how the results compare to experimental studies, right? That if you have no relationship that makes sense with experimental studies, then obviously you're not modeling the right thing. So we looked at some basic things like from the literature, from the studies that I showed you previously, and a couple from Sommer and Kammer, they measured the ratio of the multi-threshold for monophasic versus biphasic pulses. It's known that monophasic pulses have higher thresholds, and monophasic versus half-line pulses, and the threshold is approximately the same. So this is the experimental data here. And these bars are actually standard error, so it's actually very narrow bars. Really, the appropriate thing will be here to look at standard deviation, because you want to see how it varies across subjects, which will be wider bars. So anyway, in red is a very simple model of the neural membrane that assumes a linear time constant. It's a basically simple integrated fire model that people have used, and we have used in the literature, to approximately model the effect. And you can see that that model goes in the right direction, but it really does not give you the full extent of the difference. It's actually quite, quite bad. Now with our model, it gets much better. So it's quite already within this actually pretty tight standard error bar, and this bar does vary depending on exactly how you define your region of interest in multi-cortex. The bottom line is that we're starting to get better estimates that match what we see in the literature. And now, I think it gives you a very interesting insight, an important positive insight into that phenomenon of increased latency of the multi-vocal potential that has been known for the anterior-posterior monophasic pulses. So we can model that. What you see here is the cross-section of the hand knob, right? The hand knob in multi-cortex. I think people like Mark Halletti have dedicated probably the last 30 years, studying, this area very closely, the multi-cortex. So the hand knob is the area that gives you this finger twitch. And if you take a cross-section, you see this is the anterior direction, this is the posterior direction. So this is the, the hand knob is in the precentral gyrus. So here we have simulated these three different waveforms with two directions for each. And again, we can simulate the threshold for each layer in each location of neurons within the layer. You can see the 2D slices that come from that, that plane that goes through the hand knob. So this pulse gave you the strange long latency. So what's happening there? So notice what's happening in the gyral wall versus the gyral crown here, right? This is the gyral crown, this is the wall. And why these particular areas you see like these are the areas that control your finger, the FDI muscle that's recorded here. So you can see that when you reverse the direction, you can go from posterior anterior to anterior posterior, the activation in the crown remains by and large of the same, approximately the same. But you have dramatic change in the activation in the wall of the gyrus. So that's a very notable difference here when you change the direction. The field distribution is exactly the same. You're just changing the direction of the field. And that does not happen with the other pulses, which are more bidirectional, right? You see that, yes, there is a bit of a change, but still, you don't have that kind of, when you consider this relative to this, you don't have this dramatic effect anywhere else. So, that's interesting. So, does that make sense in terms of the neuroanatomy of the hand knob? And it turns out that it actually makes a lot of sense. And you can see that by looking at this beautiful figure from Rateloff and Strick, in a paper from 2009. This is like a flattened image of the hand knob in monkeys, and this area here, this is the wall of the gyrus. And this area here is the kind of lip of the, or in the crown, the lip of the crown and the crown of the gyrus. So these dots here tell you where the neurons that directly control, the output neurons that control the FBI finger are. And then here with microstimulation, they map stimulating what neurons gives you a twitch in the FBI, in fingers, generally. Right, so you see that the output neurons are here, but there are other neurons in the crown, in the lip of the crown, that still transsynaptically activate the finger. Well, right, that there we are. So it seems that from what we have seen with the monophasic pulse, in one case, when we stimulate the wall, we have more direct outputs, but when we stimulate the crown here, and the field is stronger here, we have indirect output. We have indirect stimulation, which is transsynaptic. So it's a beautiful picture that very well matches what this understanding of the neuroanatomy. So this is an example of how the simulations can really, combined with other pieces of information, can help us understand what's going on. So our time is quite limited. So I'm going to briefly talk about a technology that we have developed called Controllable Pulse Parameter TMS, or CTMS, that allows us to shape the pulse waveform more dramatically than you can with conventional devices. So it generates these more rectangular pulses. You can control the pulse width, the pulse direction, and in a more expansive and continuous way than in conventional devices. And the idea is that it can allow you to explore these effects waveform more extensively. And this is the family of CTMS devices that is developed, and now there's a commercial device sold by Rogue Research, and actually I just saw the device that, the CTMS device that is here at the NIH, and so you have one if you want to play with one here. And other labs several other labs have gotten that as well. So it gives you a lot of flexibility of waveforms if you want to experiment with that. So with that, you can do things like strength duration curves, which you can do with conventional devices. You can vary the pulse width, and you can get multiple thresholds of different pulse widths. So again, here we ran our model also with these different pulse widths, and the red curve is the strength duration curve that we got from the model, and the black curve is what we got from the experiment. Now, the overall amplitude we individualized, so we normalized so that we can see the curvature, and the curvature matches really well between the model and the experiment. We have also run our at Duke, our first study with repetitive stimulation with different pulses. One of them is conventional, and three of them are TMS pulses. So I won't go into a lot of details. The point is that we got different amounts of inhibition across the pulses when we changed the pulse shape. So again, the TMS pulses are doing something, and when you look at, again, the possible explanations, you see singular effects here. We got these pulses that, again, have more unidirectional character, and we have them in different directions. And again, when you simulate them, you see this phenomenon of, differential recruitment of the gyral crown versus gyral walls. So basically the same kind of phenomena that again results in this difference in latency when we measure these pulses. And interestingly, these are biphasic magnetic pulses that produce more unidirectional electric fields because you can really play with the rise and fall time of the magnetic field. We have published quite a bit on that, so I don't have time to go on that. And also we played with whether we predict well the thresholds that are that we got for these different pulses. So again, compared to the simple model that we have previously used and others have used, which is the linear membrane, which is in red, you can see that our models, more realistic models, which are the green bars, are closer to the actual data, which is in blue, compared to the red simplified model. And it's generally within the standard deviation of the results. So again, it seems that we are having a tool that's better predicting what's going on. And interestingly, these results are sensitive to exactly how you define your region of what neurons you consider. So there's more subtlety there that we have to really explore carefully. And finally, just a quick advertisement. We're not just playing with models, we're also doing actual recordings of actual neurons in the brains of monkeys, rhesus macaques. During TMS, we have specialized equipment for that. And we have these beautiful pictures of what happens when you deliver a TMS pulse with different intensities, right? These are stronger pulses as you go up. And then you can see how, obviously, low intensity don't have much of a response. And for the TMS pulse at time 0, when you have high enough intensities, you have this, rapid firing immediately afterwards, and then a silence period, which is actually known from also electromyography MEP recordings. And we can look at we can identify the firing of different neural elements, axons, and inhibitory and excitatory neurons. So it really gives us an exciting window into what actually happens. And it is that we're coupling that with our modeling to develop better models. So in conclusion, even though my talk is probably not the usual thing that you see about multimodal techniques where you combine TMS with fMRI or combine TMS with EEG and use that to study the brain, I can show you some less kind of obvious ways in which imaging and electrophysiology are now getting more and more important used together. And one important fact is using MRI as a technique that gives us understanding of the structure of the brain, and then we can use this structure in sophisticated models that can allow us to understand better the mechanisms of TMS. We're also then developing tools like quiet TMS and more focal TMS that can be used in MRI scanners or with EEG, and both of these can have fewer bulk kind of artifacts and also ancillary effects, unwanted effects in brain stimulation during stimulation and imaging. So, for example, quiet TMS, I think that would make a pretty dramatic difference in both TMS fMRI and TMS EEG, since the auditory component is major in both of these. And in addition to that, we can kind of elaborate these MRI-derived models with populating them with neurons that are actually originally derived from stimulation electrophysiology recordings, and we can also do TMS with neuron-level electrophysiology to understand what actually goes in the brain and use that to form our models. So thank you for your attention, and I'll be happy to answer a question or two.
So if any of the participants have questions, you could put them in the chat window. I wanted to ask, how does the modeling and the finding of the crown versus the wall being simulated explain our TMS that you showed very much greater one direction inhibition actually?
The question, that would be quite a wide leap to say anything about that yet. We are also doing work on creating cortical column models where you try to wire everything, and then you have to add synaptic plasticity to that, and then we can start to really understand that better. So we're not there yet. So we can't, I'm not even trying to explain that other than saying, look, we are starting by stimulating different neural types in different proportions, right? And you can see how that could then ultimately lead to different neuromodulatory effects. But exactly how that happens will need more circuit level models. And again, that's something that we're interested in and working on.
The comparison you did with the simple linear, the realistic, and then published, was that just to the published normative data, or is that using the actual MRIs from those published studies?
So this was basically, it's essentially comparison of multiple thresholds, and there you take so just to repeat the question is whether we have this, when we have comparison between simplified models and measurement data and our more complex models for thresholds, we're mostly looking at ratios, because in terms of the exact absolute threshold, we're still, it can be off by some percentage, so that's kind of, this means that you have still some factors to tweak, in the model. So what's more important to us right now is to look at the ratios, because it shows difference, and there we normalize, what we did is we normalize something. For example, we normalize the stimulation for one pulse, and then we look at the differences for the other pulses, which is equivalent to looking at ratios.
Sure, but you're comparing to the published normative data, right? You're not looking at each individual MRI and then reconstructing that and then looking at that ratio for that.
Okay, yes, no. Okay. Right. We're not looking at individual MRIs. No, this is, right now it's just one CAD model, one standard CAD model, and that's why I'm saying that the proper error bars on the data is the standard deviation, which gives you the variation across subjects, because this is one subject who's not even part of that study. So, still it's, it has to, it's one of the things where, again, we're building from the ground up, right? So okay, let's make better neurons, and then, and then at some point you can start putting them in individual head models. That's actually straightforward. It's just a matter of acquiring the MRI, from the same subject that you did the experiment with, and then populating and doing all these calculations. So that should be doable at present.
All right. Add a little bit more. So you're showing the single unit EFIS. Have you looked at how your predictions for the layers firing compare with the EFIS data? The monkeys, obviously, you need to do a monkey head model with that, but.
So we are, with the electrophysiology, that unlike the simulations, that takes a long time to collect data. So you see here that we have 287 neurons. This is basically 287 work days plus of these studies. They very, very complicated. So we are very interested in trying to get information of actually where exactly the electrode is. And you can, if you sacrifice the animal, you can tell that, but these are monkeys that are used for many years, so it's not like you do one experiment and sacrifice the animal. So it's a bit tricky to establish where exactly you are, and we're looking at techniques that can allow us to identify more specifically exactly where you mark there, and then we should be able to get such information. And also, we're using this relatively simple but robust tungsten electrodes that are just single channel, and from that, with data analysis, you can extract up to like 3 neurons at best by interpreting the amplitude of the waveforms. But you can use also, more complex electrodes that have multiple contacts. For example, going in depth, getting multiple contacts across layers, and that also may be an approach going forward. The issue here is that the throughput of these studies is quite challenging to get high throughput, but we have an H1D, so we're doing well, and we'll probably scale up the effort to get more parallel animals so we can run more conditions and explore more questions at the same time. And this is the monkey work I want to mention is done in the lab with my collaborator Mark Summer to do. I believe there was a question online.
There is one. It says, how sensitive do you think these results are to gyral geometry, and if you have validated these simulations using more than one model?
So, no, we have not really looked at multiple models at present, although that would be really straightforward. So in terms of sensitivity, we have not evaluated that quantitatively. Qualitatively, I would think that, yes, it will have an effect, especially at some level. but that may be more at the level of the electric field rather than, you know, the neural, because you still have, right, unless you have some really unusual subjects with some differences like thickness of cortical layers, you'd expect that structure is quite consistent. So then most of the difference will result from the cortical geometry that affects the electric field. So it's not just the kind of this gyrus, but it will be more like the gyrus and the surrounding sulci and its particular height and the overall, you know, geometry of the conductive layers that we have around it. So that can be explored. I think that the main effect will be from the fact that, for example, in the hand knob, from the fact that people have very different hand knobs, and in some MRIs, some subjects, you don't even exactly see a knob, you know, at least on the surface. It's kind of hard to identify. So I think this variation can result in differences. And it will be very interesting indeed, as David here was suggesting, can we do that individual MRIs? And we can do that. And that would be a very interesting study to correlate actual experiments